and I'm here to give you a talk on non-native fish species. So, a large portion of the world's biomass is composed of non-native and invasive species. Therefore, the study of these non-native and invasive species, particularly their biology and ecological physiology, is very important to understand not only how they are able to become so successful in their introduced environment, but also the effects that this has on the native fauna and flora. So in my study, I concentrated on two non-native fish species found in Florida. So what is the concern about these non-native species? Well, first off, they are able to spread disease and pathogens. They're able to lower water quality, affect the fish community structure. They can change the trophic dynamics or the food web that they're thrown into. They're able to create hybrids with the native and also other non-native species. And lastly, biological homogenization can occur, which is just a fancy way of saying the overall biodiversity is decreased in that area. So I wanted to share this quote with you as it discusses the importance of studying these invaders. It states that the species invasions provide experiments that can be used to better understand the natural world, including our understanding of application of high problems such as extinction, ecosystem functioning, and the response of species to climate change. So, what characterizes a successful invader? First, they must be able to adapt to the environmental conditions that it is introduced to. Next, they must be able to reproduce quickly to establish a population as fast as possible. They are competitive with the native species for food as well as space. They are predatory on the native species. They are able to avoid predation themselves. And lastly, they are generous feeders, or pretty much just garbage disposals, in that they feed on almost anything. And my study kind of goes against this, as I have an example in the pike killifish, which is seen on your left here, as it has a specialist feeding mechanism which goes against non-native science. With the pike killifish, you can see here, the close-up of the head, I want you to notice the jaw structure. We have elongated jaws and teeth specifically made for piscivory or feeding on fish. In comparison, we have the Mayan cichlid, and this is a generalist predator and has the jaw morphology that you will see in most common fishes. They can feed on anything from algae, crustaceans, to mollusks, etc. Now I would like to draw your attention to these two maps of Florida, and the range of temperatures is the average temperatures in that area in Florida. I use these to create my temperatures, the temperature range in my study, which is from 20 degrees all the way to 30 degrees Celsius. Now, the coloring that you see on these two maps is where these two species have been found in the state. The very interesting part about this is that both of these species was introduced into the same area of southwest Florida. The big difference between the two, the generous Mayan cichlid was introduced 26 years later than the pike killifish. And yet, as you can see on the east coast here of Florida, they have a much faster northerly rate of spread than the pike killifish. This leads me to my rationale, which is that the pike killifish is as successful as the Mayan cichlid in its non-native environment, despite having a specialized feeding mechanism. This suggests a paradox in the non-native science. Secondly, what is the ecophysiological basis of the apparent difference in the rate of spread between these two non-native species? This brings me to my hypothesis, which is that at a given temperature, 20, 25, and 30 degrees Celsius, the prey capture kinematics and behavior will differ between both of these non-native species. And secondly, that these kinematics and behavior of each non-native species will vary across those three temperatures. So why use feeding kinematics? Well, kinematics quantifies movement and behavior, and I used the prey capture event of a fish to study this. In the top diagram here, we have the three phases. And the first one is preparation, where the muscles in the jaw begin to tighten or compress, And as that mouth opens, we have a negative pressure created in the buccal cavity or the mouth of the fish. As it moves to food capture, we have a giant explosion in the mouth. And as this occurs, from that negative pressure, water is being sucked in as well as prey. And then it finally moves into the mouth closing and swallowing its prey. So as you can see, there are many bones and many muscles in this process, and these are used together to quantify, I use the study of this to quantify the physiological performance in these animals. So for my data, I collected 4 to 6 individuals from the wild. I acclimated them in the lab and trained them for high-speed filming. This is my camera system here. I used a red light camera system. They were fed live gambusia, which is that minnow you can see on the left. As I said before, I used 3 experimental temperatures, and 4 films were analyzed per fish at each temperature. So here are 2 representative films from my study. I just put these in here for you to get an idea of the type of films that I was working with and where I got my measurements from. So on top we have the pike killifish, which is the specialist, and on bottom the mine cichlid, which is the generalist. Now I want you to remember the differences in the jaw morphology that I talked about earlier. Let's click the actual film. Put the like. Now keep in mind, this is at extremely slow motion compared to real time. All right, so once all these films were taken, they were transferred to a MaxTrack software for analysis. Now in this software, I had a total of 46 films. From those 46, in each of those 46 films, I measured 9 kinematic variables. For each of these variables, I took 3 measurements per. And this, I took the average of those 3 measurements to make sure my values were as accurate as they possibly could be. So my first 2 variables were maximum gait and time to maximum gait. Next were maximum high rate depression and time to maximum high rate depression. This is measured from the bottom of the eye to the floor of the mouth. Next we have cranial rotation and time to cranial rotation. This is the angle that is created when that jaw is opening and the head thrusts upward. After that is maximum lower jaw angular displacement and time to. While the head is being thrusted upward, simultaneously the lower jaw is being forced down and that angle is being measured. My last variable was approach velocity, and this was measured from the back of the eye of the predator to the back of the eye of the prey. This was my first distance. As the predator approached the prey, I stopped the film at the frame right before the prey entered the mouth. The distance between the eyes was then again measured. My formula provided me with the value as d1 minus d2 over time. This is an example of my excursion or displacement kinematics. With this, I have maximum gait on top and maximum high rate depression on the bottom. The orange bars represent the pike killifish. The blue bars, mine cichlid. And as you can see in these graphs, we have a higher value for the pike killifish at both distance variables across all 3 temperatures. Next, we have an example of my timing kinematics. And with this, time to maximum gait and time to maximum high rate depression was measured. For this, the opposite was true, as the mine cichlid's values were greater than the pike killifish. So what this means is that the pike killifish, for say max gait, is reaching a greater value for max gait. Not only that though, but it's doing it at a much faster rate than the mine cichlid. And lastly, an example of my angular kinematics with lower jaw displacement. And from this, you can see across all 3 temperatures that the values were very constant within species. So I forgot to warn you about this. There's a lot of numbers on here, but don't worry, they'll be synthesized in the next few slides. From this, I just wanted to show you my 9 kinematic variables on the left, and up top, my 3 temperatures used. P stands for pike killifish, and for Mayan cichlid. These are just the means and standard error values that I got. So from those numbers, I ran a 2 factorial nanovar using species and temperature as my factors. From this, I got 2 very interesting results. First off, the kinematic variables are significantly different between species across temperatures. So across the board, the pike killifish was significantly different from the Mayan cichlid. Secondly, for each species, the kinematic variables, except approach velocity, are not significantly different across temperatures. The temperature didn't seem to play a big role in the actual prey capture event, just in the movement of the fish. So to summarize my results, I'll bring you back to my hypothesis. The first one was that at my 3 temperatures, the kinematics and behavior would differ between the species. This was supported as the excursion and timing variables were shown to differ. My second hypothesis was that the kinematics and behavior of each non-native species would vary across my 3 temperatures. This was not supported as the prey capture kinematics did not vary throughout. So the big picture of this study is that these results indicate that the specialist and generalist non-native fishes have similar prey capture responses to temperature. And lastly, the difference in rate of spread across Florida may be driven by factors other than this physiological tolerance to temperature. And that concludes my study. I would like to acknowledge my advisor and mentor, Dr. Ralph Turingen, my lab members, my chapter president and advisor, Dr. Michael Grace and Stephanie Visser, and of course, Dr. Carroll, who is our last-minute fill-in advisor. He got us here in one piece, so let's hope he has a repeat performance on the way home. And lastly, Sigma Xi, who provided funding for my